Welcome to Saitechinfo channel, my dear students. In this tutorial, we will be discussing about process of recombinant DNA technology. This technique is applicable in bacterial, plant and animal cells. It has four stages. The first one is isolation of genetic material, that is the DNA. Then cutting of DNA at specific locations. And then insertion of recombinant DNA, that is called simply rDNA, to the host cell. The host cell may be a bacterial, plant or animal cell. Then obtaining the desirable foreign gene product. So these are the essential four stages of the process of recombinant DNA technology. Let us see the first one. First we need to isolate the DNA. So isolation of DNA, where we need to restrict certain enzymes. What are enzymes? These are all biocatalysts. So we need to avoid certain enzymes to get activated. And the DNA should be as pure as possible. So it needs to be very, very pure. Then it should be free from some macromolecules, so that should not interfere. And then these things are feasible by treating with lysozyme. So lysozyme means it is the antibacterial enzyme produced by animals to form some immune system. Second, cellulase. Usually the suffix ase represents the enzyme that breaks the respective compound. For instance, protease means it is the enzyme that breaks the proteins. Similarly, cellulase means it breaks that is the enzyme that breaks the cellulose, that is the plant cell. Similarly, chitinase, that is in the fungus you have got some glycosidic wall, and that should be broken. So this is the enzyme which is responsible for breaking down the glycosidic walls of the fungus. So likewise, we need to treat the cell, the DNA, with all these lysozyme, cellulase, chitinase in order to get the purified DNA. And then RNA should be removed by a ribonuclease. As I already mentioned, ACE is the enzyme that is responsible for the cleavage of the respective compound. So here when I say ribonuclease, means it is the enzyme that breaks the RNA. And then proteinase, as I already told you, proteins are broken by the protease. And then further it is purified. Now you have got the purified DNA in the precipitate form, and that is blended with chilled ethanol. Ethanol, that is ethyl alcohol, C2H5OH, that is the spirit. Now let us come to the second part of it. The second part is cutting the DNA. So we need to cut the DNA at specific locations. Then the restriction enzyme should be digested by incubating purified DNA with restriction enzyme. And the third one is agarose gel electrophoresis. So it's a kind of electrophoresis, means that is electrophore, the charged sphere, the charged compound that undergoes the cleavage. So that is called electrophoresis. For example, you have got electrolysis. Electrolysis means a cleavage of electrolyte. Similarly, the charged bodies, when it is subjected to electric current, then it is broken into cataphore and anaphore, and the movement of such kind of charged bodies can be called electrophoresis. So using this agarose gel, we are able to check the progression of restriction enzyme digestion. So we need to further stop the progression of this enzyme digestion, and we need to take care of the gene of interest to us. So that is called GOI, that is gene of interest. So that is taken from the source DNA, and the cut vector is mixed and ligase is added, so that we are able to get the recombinant DNA. So this GOI, that is a gene of interest, is further amplified using PCR. And then insertion of rDNA, recombinant DNA, into the host cell. We need to insert the rDNA into the host cell or the organism. Here we have got the vector DNA with the plasmid DNA, is it not? And that is the plasmid DNA. The vector DNA is the plasmid DNA, and the alien DNA, that is the foreign DNA that carries the what? Our gene of interest. And both are cut with some restriction, the same restriction. The endonucleus to produce complementary sticky ends, so that it can be tied up, it can be made to mix in a proper way. So vDNA, that is the vector DNA, and aDNA, that is the alien DNA, which is nothing but the foreign DNA with the interested genes. So these are joined by the DNA ligase. So this is called the annealing. It is the process of mixing of vDNA and aDNA using DNA ligase. So that is called annealing. That process is called annealing. Annealing. Okay. Now the recombinant DNA, that is chimera, that is the product that is produced. So the chimera, that is the recombinant DNA, is perfectly produced. And now we have to further, that is you should know something about the eukaryotic genes. So they should not function properly when cloned into bacteria cell due to introns. So there should not be any interference with the growth of the cell, and that is why now the DNA made from mRNA by reverse transcription, that is, or by synthesis, and the rDNA, now it is inserted into host bacterium, that is called transformation, and we can use calcium chloride solution here. Then finally the rDNA selected in such a way using the selective antibiotic in the culture medium. Then we should have the desirable foreign gene product. So that is the gene of interest, is it not? So this rDNA, the recombinant DNA, is transferred into the host cell with the multiplications of foreign DNA, is it not? And our aim is to produce the desirable protein only. So the recombinant protein encoding a gene in a heterologous with the different media, heterologous hosts, and we need to maintain the optimum conditions for cloning the GOI. What is GOI? That is the gene of interest to us. Now we need to induce the expression of the target gene. Then finally we can produce it in the large scale, and it can be taken packed for marketing purpose. Now let us check the schematic diagram. See, here you can see the nucleus with the nucleus of the donor cell, and it has got the foreign DNA, is it not, fDNA? So we have got that that is double helix DNA. Are you able to see that? The ladder like DNA with the foreign DNA. And here the bacteria cell. So that is the host, is it not? Where that is you have got the plasmid and what do you call the bacterial chromosome is present there. So the bacterial cell with its plasmid and the chromosome, and that is the vector DNA. We need to take the vector DNA, and here in this case now we have made this as a sticky DNA. So sticky alien DNA, that is foreign DNA, and this is the vector DNA. So that one is alien DNA and this is vector DNA, and the process of mixing. Okay, so we are mixing this, is it not? So this mixing is called the annealing, is it not? And after annealing, we are able to form a good rDNA. Then rDNA is made to, we have to seal, there will be some gaps, you can find here, there are some gaps. Okay, so these should be, that is completely sealed using the DNA ligase, is it not? Now we will go to this part, that is here. So we have got the nucleoid and plasmid free from, plasmid free bacterial cell. So there should not be any plasmid. It should contain the that is chromosome. And here after mixing it, that is the transformation takes place in this case. Here. So let me see. Are able to see that? Now after that we are able to, that is get the transformation of rDNA, and what is the chemical used for that? The chemical used for this is calcium chloride solution, don't forget that. And then the rDNA and the host bacteria with this rDNA, recombinant DNA. So that is available here, and the clones are produced by the multiplication. Then all these clones are engineered. So these clones of engineered bacteria, okay, so perfectly designed bacteria to our requirement. So that is kept in a fermentation tank, so that in that medium it will start growing. So now we have to downstream everything and further purify, then we can pack it and market it. So this is the overall schematic diagram of the recombination, the process of recombination. So if there is any doubt, you can ask me, you can put it in the comment box. Thank you for watching this video.